Okay, welcome to our GRG online meeting. Today is Saturday, October 9th, 2021, and we're going to get this party started here. I have several interesting topics. There's one thing I want to talk to you about initially and kind of float it by you, certainly get your input. This appears to be something very, very worthwhile I've been working on for a few years, a couple of years at least. And I'll be talking to you initially about how we intend to help the most innovative researchers and companies accelerate the creation of exceptional aging intervention therapies to facilitate the end result we all seek. And that end result is, of course, a great many more healthy years of healthy living. Now, specific proteins have been reported in multiple studies to change their expression levels with age. So the protein clock is an innovative analysis system to measure them. The most important use is to compare baseline and follow-up results to measure the effects of age management therapies. And the expectation is that older people who reset their protein clocks to a profile associated with younger people will be healthier and live longer. And in some ways, it's like measuring DNA methylation. And, you know, the protein clock can be useful in evaluating biological age, but in my experience with any clock, it's, you know, what's really most important is tracking improvements after a therapy. So it's innovative and a much needed marker. And this could be a game changer. So I now have a working prototype.
Could I just make one comment?
Real quick.
It's much further down the road than DNA methylation. That's the nice part about it.
Well, thank you.
It's methylation of, I mean, I don't mean in usage, I mean in what it says about you, a person. Yes, and it involves both the methylation of epigenome of the DNA plus the epigenome of the non-DNA parts. There's factors in there that affect the DNA expression. So it's really much better, I think, if it can be really done.
Well, thank you, Paul, because in my estimation, it's much closer to measuring the end result we seek, which is function, but really, you know, what's going on in the body. And you be the judge. We're going to dive a little deeper this time. You be the judge whether it can be done. I'm going to show you. So I'm going to be starting a campaign and submit it to Lifespan.io, and it's to take it to the next level. And that is to evaluate and verify low variability among samples, and that the process can be systematized to work in a larger scale production mode. Okay, now world-class scientists, Benoit Lahallier, Tony Weiss-Correa, Maxime Shokarev, and Adib Johnson.
Thanks.
Developed the original protein clock and wrote a paper about it. So let's take a look at the original paper, and I'm going to share my screen.
John Kramer doesn't want to interrupt you, but he has his hand up for a while.
Okay, John, go ahead.
Yeah, I have a question. It seems to me that different cells are going to express different proteins, and therefore it's going to be important whether you get the sample you're analyzing from the blood or the urine or the or saliva or cerebrospinal fluid or whatever. And so I was wondering whether you're planning to cover all of those bases or only use one sampling technique.
Yeah, well, you're jumping ahead a little bit, but I'll answer your question now. We're going to use blood. Blood touches everything, and the test system we now have available is only available for blood. So that would answer your question. And let's see. Yeah. Okay. So here's the original clock. Determining human plasma proteins, et cetera, et cetera. You could read that. Let's scroll down a little bit. They talk about some of the things I'm talking about here. The abstract is very useful. But let's go down a little bit to, these are just 6 of the sample proteins associated with age. Their quantity in blood either increases or decreases with age. And a little farther, this is the aggregate, and this is the learning set that they use, and this is the result of the test set they used, again, on the original paper. So, Benoit, the lead researcher on the paper, he was kind enough to help me develop a practical clock based on his work. And he's a very busy guy and real smart. He's now director of data science at Alkahest. So I'll describe the process for you a little bit. It starts with drawing and processing some blood. Freezing it at -80, then sending it off to SomaLogic to have their proprietary SomaScan panel run. And the panel evaluates over 4,500 proteins. So data is extracted from the resulting digital report, then a computational process is applied to the 491 most important proteins associated with aging, as identified in the paper. So for each subject, the protein is multiplied by a coefficient to arrive at a score. Then these scores are totaled to arrive at a final score. So then a lower number represents a younger biological protein age. And by the way, there are other ways to apply this technology to evaluate and help with specific diseases. So for our next level project, 15 samples will be run. And so now we'll go a little deeper into the technology, and I'm going to move rather briskly on this and share the screen again. So here is, we're jumping to the end result with results and age, and it's plotted on a curve, and we see there's a roughly linear relationship, but with some outliers, which are to be expected, okay? Okay, this is what's called an ADAT file. It's the SomaScan Outlook. It comes in a text file, but I transposed it into a spreadsheet. It's much easier to work with as a spreadsheet. On the top is like administrative housekeeping stuff. Then we scroll down. There was like some blank space here, which seemed peculiar to me at first. But then we go down, and I've yellow highlighted some sample ID column. So now we're starting to look like subject information. Then we scroll over to the right, and here's age and sex information. So now we're getting into something useful. Then to the right, a bunch of numbers, but if we scroll up again, we see what looks like proteins. So CRBB2, that's what, beta crystalline B2, CRAF, that's apparently, yeah, apparently there's a whole family of them. And we go down and we scroll to the right, and we keep scrolling and scrolling and scrolling. And what this SomaScan returns is actually 4,884 protein values per subject. So here are just a couple of key pieces of information that Benoit sent me so I could devise the calculation. Here is the result of the very first one I ran, a 55-year-old male, and the proteins, their values multiplied by a coefficient, and then the total. So his score is a little over 17,000. Okay, here are the first 64 that I ran with subjects of different ages. Okay, and here again is the end result, which is the plot. And, you know, we want to see, what's important to me anyway, is how innovative aging therapies shift these measures younger towards youth. And, you know, my interest is the practical application in this to apply it to aging intervention therapies. So after successful completion of the next level, a proof of concept verification, the new organization, Aging Metrics, will initially make it available as a research grade test, and maybe later as an FDA approval, but initially to accelerate the work of the most innovative researchers and companies working on therapies that'll get us a great many more years. That's the point to all this. So it'll be offered at low or hopefully no cost to the most innovative researchers. So some of my specific interests that I hope parallel with yours include epigenetic reprogramming with therapies like Yamanaka factors and others, and gene editing, senolytics, removing pro-aging factors from blood and tissues, and mitochondria therapies. Some of the companies that will be offered to include Turn Bio, Life Biosciences, Nugenics, Ocean, Altos Labs, BioViva, Integrated Health Systems, Shift Biosciences, and Iduna Therapeutics. And some of the scientists include Rodolfo Goya, Juan Carlos Belmonte, George Church, and James Clement. And there are others now into the future. Really. So many, so little time, and we need more time. That's what this is all about. So since we're in this for more healthy years, that's the end result we seek. This will be offered, again, I say, at low or perhaps no cost. I don't get a salary out of this. So that's pretty much it. And I hope this meets with your personal goals. And so what do you think?
Okay.
Yeah, I have a question. Yep. Arena Conboy, in her paper on plasma dilution, mentioned some kind of thing about protein testing that sounds very much like this. I wonder if this is the only game in town, or whether there are other people or groups or whatever that are using protein as a clock or as an indicator that something happened when they did some intervention.
I believe there are others.
Mm-hmm. Are you using a biochip to do the analysis? Well,
it's we can take a look at SomaLogic website and some of their white papers, but you know, I personally don't have a lab that's going to do this.
But you're doing 4,884. I don't think there's a test tube for each protein, is there?
Oh yeah, you have to draw 4,800. No, it's amazing the technology. It's a small sample of blood, just one tube. The processing.
It's not on a chip though. It's something else, wells or something.
It appears to be wells. Yeah, we can dig deep into that, maybe privately or in a separate meeting. If you're interested, I mean, I'm
just a pedestrian. I don't know how to do such things, but
yeah,
I'm used to physics labs, not wet labs, and so.
Johnny, do you know what the cost is to run a single sample and get the?
Well, yeah, the cost for us, they've been working with me. And, you know, normally they require like over 40 samples, but for our test, I have a real good relationship with them. They're going to run just 15. And for 40, I think the cost is something like $650 per. Okay, for this 15 sample, you know, it's going to be $700 per sample. And I know that's pricey compared to other things out there, but it's value versus cost. And
yeah, the problem is that you'd like to do this more than once. You'd like to do it and then do an intervention and see if it changes, and then do another intervention or several interventions as a function of time to see if the effect was fading away or whether you had a permanent effect. So
well,
that involves a lot of testing.
Yeah, and a lot of money. Hopefully the cost will come down in the future, but for now it is what it is. And that has actually been one of the obstacles that kept me from moving on this about a year ago.
Mm-hmm. I have a question.
And also, if you want to be.
I have a question related to actually what I thought of when John Kramer asked a thing earlier about what cell type, cell types and so on. The question relates to that. And if you're just doing blood, then there's a lot of proteins go up and down in the blood with respect to age, but some of these may be things that need to go up and down or go up and down for the purpose of helping the body deal with age, you see? So, you know, if your therapeutic purpose is to sort of set these proteins back to young levels, that may not be good for you. And that's my point. So you need to look at the whole overall picture a lot. Okay.
Yes. That is something I've thought of, probably because you brought it up when I briefly touched on this in one of our last meetings, and it needs to be considered.
Yeah, I think it's worth investigating whether you can do something similar with urine or saliva, because drawing blood is a significant barrier if you want to do something on a large scale with a lot of people.
Well, yeah, there's that. Plus the processing, the phlebotomist, you know, this is within the skill level of many phlebotomists, but first, they according to SomaLogic, Serum produces slightly more, less variability in the results than plasma. And the process is, you know, there's a post-draw processing where they have to spin it down. They need not a really special centrifuge, but one that some phlebotomists don't have. So we'd have to deal with that.
As an ignorant physicist, what's the difference between serum and plasma?
I believe serum has, oh God, yeah, I know I should know this, but it's one of the components is removed. I believe it's the buffy coat or the immune cells. Any real biologists here know the answer to that? Okay. All right. Now, I believe, Robert, did you have some questions? No? Okay, I guess not. And let's see, I see in the chat, is that the chat? Nope, no questions in the chat.
Well, I have a point about the cost or the difficulty that John Kramer alluded to of getting the tests and so on, or the applicability to various people and so on. It may not require, and that's the question is, how much blood does it require? It may not require a lot of blood, it may require just a finger stick.
I'm here, I'm listening.
And it also may just require blood on a blotter, a filter paper, or whatever it is called, which is the tests I just did for Ginfinity, which I'll be posting soon, my results, comparing NM with NR, were required only a little finger stick and drops on a card. And that's much easier than getting a phlebotomist to draw a tube of blood. So the question is, how much blood do you need for these chips? I guess that's the question here.
Yeah, it requires a tube, or actually less than that, but it's drawn into a tube.
Well, Ginfinity has actually two methods of doing these things. One of which, in fact, is Aging SOS. It doesn't require a phlebotomist. You can actually drip the blood into a tube from your finger. And so maybe that is enough blood. He gets sort of a few grams of blood that way, I guess. And that's all he needs. So maybe that would be enough for this test. And again, anybody can do that who doesn't mind the pain and the squeezing of your finger to get a bit of blood out.
Yeah. Well, for this test, some processing is required. It's just necessary. And then freezing on -80. And I just want to stress that initially, I mean, in the future, who knows what the future will be, but initially, this whole, what I'm doing is to facilitate the work of the most innovative researchers. So I'm going to be very selective. This is not a mass marketing kind of thing. It'll just be offered to those most innovative scientists and companies working on therapies that, we can expect will get us, when fully developed, a lot more healthy years.
Johnny, is there a subset of the proteins that you are looking at with the present analysis that have been documented to be problematic, and that you could narrow and look at several different subsets and see if you are able to optimize the values that you get before we actually know, as Paul mentioned, whether it's good or bad that these go up or down? So selectively choosing those where we already have some indication of positive or negative effects.
Well, there are 491 that are used in this evaluation that are associated with aging. And there are also other proteins associated with specific diseases and disease conditions. And, there are, I think the number is 9. It's about 9 have been identified that are particularly useful in and associated with aging. And, the presumption is, and I know it may be a bit of a leap to suggest, what Paul pointed out, that, okay, are certain levels of certain proteins needed at a particular person's age to support what's going on in their body? And if you were to do something to reset them all, would that actually result in worse health and reduced longevity? And we certainly don't want to do that. So that's why I'm careful to say we're taking this to the next level, and that the expectation is that a better protein profile will be healthier for a person. And this would be compared, I didn't mention it. Thanks for reminding me. This would be compared with other clocks, DNA methylation, other inflammation measures, cognitive tests, functional tests, grip strength, 6-minute walk test, that sort of thing. So it would be a composite result. And, like I'm saying, this is going to the next level. I don't claim to have all the answers here.
I would think that
I do have a question about the coefficients that you've chosen for each of the proteins. I noticed that they were different for different proteins to develop the final score. How did you choose those different coefficients, and why are some of them so much larger than others?
Well, actually, the team that wrote the paper, Benoit was kind enough to send them to me and help me over the hump with some critical parts that I couldn't personally extract from the paper. They came from his research.
They were arrived at by some kind of regression technique that selected out the proteins that were best correlated with age. Is that what's going on?
Yeah.
And some of them go up with age and some of them go down with age. So some of them have positive coefficients and others have negative coefficients. Is that right?
Correct. And as Cliff pointed out, some of them increased or decreased a lot more than others.
What about daily? I would think that as methylation ought to be more or less rock solid, but I would think that proteins fluctuate on a daily basis in the blood as they would be different when you're sleeping than when you wake up, than in the middle of the day, than in the evening. And so how do you deal with that kind of variation?
Oh, well, the protein test, either baseline or after therapy, would all be done as much as possible under the same conditions at the same time of day. You know, let's face it, we're not lab rats, and you know, whether we're dealing with something like this or you know a standard you know CBC, C-reactive protein lab test, or this test, we try to control it as much as possible, but there's some things we can't control. And
well, the question is, how big is the variation compared to the effects you're looking for?
Well, we're hoping the variation, we're hoping for really big effects. That's why I'm only going to work with scientists and companies that are working with therapies that are going to get us big
right.
effects. You know, we can, you know, go along, you know, with what I've come to call the usual suspects, you know, which, you know, great health habits, exercise, nutrition, metformin, dasatinib, you know, and other senolytics, rapamycin, GDF11, even umbilical cord plasma, and get a few shifts in years in DNA methylation and other things, you know, inflammation, mental measures, physical measures. But we're looking, you know, for breakthroughs. We're looking for blockbusters. I would
think that you need to calibrate this by, at least for a few people, measuring over and over again during the day. They're going to take maybe 12 blood samples during different times of the day and see how much variation there is.
Oh, right. Well, in an ideal world with 700 times, you know, $10,000, we do.
Well, you don't have to do that for everybody in every test, but I think to get started, you need to know what the daily fluctuations are and make sure that they're not large compared to the effects you're looking for.
Great point. Well, here's what we're going to do. And I have a longer write-up. I decided not to take you through everything I had to say, but of the 15 subjects, at least 2 will have 2 drawn on the same day and then 1 drawn under the same conditions, same time and everything the next day. Okay, then we'll probably make that 3. So 1, 2, 3, 4, so that's 9, probably 6. Okay, so that's 6 of the samples taken on 2 subjects. And then probably for 2 more, one of them will be at the same time on the first day. Another one will be at the same time on the first day and then the second day. So I think that's 10. So then leaving room for 5 more, just samples. And I know that's not nearly enough for statistically significance, but this would be thought of as a pilot study. And like I'm careful to say, we're taking it to the next level. And if we get wild fluctuations, you know, from day to day in those that have their samples done day to day, or even wild fluctuations in the replicates that are sent on the same day, well, then we'll go back to the drawing board and see whether that, you know, whether that variability is enough to support, you know, proceeding any further with therapy that we expect to get a great.
I would think that the most stable sample would be right after somebody gets up in the morning.
That is the plan.
On the other hand, it's probably hard to get a phlebotomist to show up right after somebody gets up in the morning. So,
well, they're used to it. You know, they're used to people fasting and you know, or we'll just find one who is, you know, they're out there. And then, you know, if we really need to train one and fly him or her out, or select subjects that are in the same location and go one after the, you know, we'll figure that out. But I could show you a somologic white paper that shows their tested variability with I think the number is 2,600. They show it as being, I think 2.6% for, on average with the proteins for serum. And if the plasma processing is a little simpler, there's a little less weighting, that's something like 3.3%. In my estimation, if we're looking for a minimum of 10 or more percent, that in a larger study with statistically relevant number of subjects, that'd probably be acceptable. So,
Johnny, don't you also have the option in this, through this kind of a study, that if you see significant variability in certain of the proteins, sample to sample or time of day, that you can throw those out and reduce the total number from maybe 490 something down to a batch of proteins that don't show that variability, and then create a more robust test?
That's a great idea. Thanks. I appreciate that.
Another comment is, if money was no object, what kind of a study would you design? What would the outcomes you'd be looking for? And where would that have the greatest value out in this ecosystem of companies and products that are out there? And is there an entity or multiple entities that would have fewer financial restrictions, but recognize the value of this grander vision and wanting to get on board earlier and be willing to spend the kind of money that would take to get the numbers of samples that you need?
Yeah, I think I heard two questions there. The first one is that I would run the 15 samples, get hands-on, you know, work out the system with phlebotomists, you know, all of that. And then I would immediately offer it, ideally at no cost, to the companies that I mentioned. Again, very selectively, companies and researchers working on the most innovative blockbuster potential therapies. But and that answer is kind of the same if there were unlimited resources. Yeah, I'd kind of do the same thing, but still we need to take it stepwise and get our hands in it, taking it to the next level to, you know, to work out the kinks. I'm highly, highly confident that the mechanics of, you know, it's a little trickier than just sending a phlebotomist out, draw blood, you know, ship it overnight, but not that much of a big deal to be insurmountable. I've talked it over with phlebotomists, and it's within their wheelhouse.
Well, I think it's an absolutely marvelous concept, and I'm wondering, how are you funding the pilot study?
Yeah, I'm going to be putting together a presentation, both a written one and a video, and submitting it to Lifespan IO. Also, I'll be refining the discussion and offering. I've already offered to make it to Allison of the Foresight Institute, and I understand that Aging Analytics Agency on the 19th through the 21st, they have a biomarker conference coming up, so I'm going to offer to talk to them about it. I probably won't be hitting them for money, but hopefully, both scientist partners who would want to help facilitate this, and you know, ideally people with money who see the vision, who would want to write checks would come to me, and, you know, we'd be really off and running.
Do you know what your estimated budget is yet?
Yeah, for this, $14,000 for this initial pilot study. The $10,500 for SomaLogic, and that, you know, 700 times 15. And then money for the phlebotomist, things like shipping, and then a little bit of a hedge for unforeseen circumstances which seem to pop up like this. And I do have some other folks who, you know, if we run over budget for some reason, I'd put some, you know, a few $1,000 into it with my own limited money. And I have some other folks that that could help out. Or
is
spend
this through
yeah.
UCLA?
UCLA is not involved in this.
And are you developing any intellectual capital along the way, or is that this is all going to be public domain?
Well, it's basically public domain, but here's the deal. I would not, I'm in this, you know, for the end result, more healthy years for all of us, you know, but to that extent, I'd help anyone move that along. But I've done things before where I've helped people, and they take it, and then later they're running with it, and it's kind of like, well, you know, what's going on? What are the results of this? You know, and it's kind of like, who are you? So if I'm going to share the secret sauce, it would need to be a clear agreement that we're in this together in the future, and I'll help you in any way I can. And yeah, well, yeah, let me address on what it took to take this from that sample data file to the end result. I was in data analytics, databases, and a lot of computer programming, software development for over 20 years. And, you know, I was in the aging, doing the aging part-time because I had to work at the time until 2013. I left a real good job, but, you know, tick-tock to work on solving aging full-time. But so I'll say that extracting that data file and coming up with this process took like every shred of creativity from, you know, that I had done. So there's a secret sauce to it in terms of arriving at the calculation file and a lot of commitment. So but it's no real secret. I mean, anyone could, if they really wanted to and had the skill, could recreate the process. They could dig into Benoit's paper, and, you know, Benoit's time is very limited, but, you know, he's a nice guy. He would probably help others as time permitted. And there are others. I understand there's a open source type of clock. There's, I have a link to it around someplace, but I could never, I opened the link and I didn't see where it was or couldn't figure out how to use it. So I had my own tools and my own skill set. And I just said, what the heck, I'll use my tools and extract this and then join it up with, you know, with Benoit's data and run it. So here we are on that.
Has SomaLogic expressed an interest in maybe being able to offer something like this in the future?
Oh yeah, this is, the Soma scan is something you can buy right now. And I'm, you know.
No, I meant the analysis that you're talking here in terms of a protein aging clock.
Oh, I suppose they would. We haven't really talked much about that. Or maybe it, you know, maybe it came up in passing in some previous conversations with various reps, but they do have some other forms of processing. I think one is for diabetes, another is for PEMF, and some others, but I don't get too concerned with those, myself right now, other than to mention them.
Yeah, I think they're looking for diagnostic protein profiles that would help determine certain disease states, which is exactly what you're looking at here for aging. So, well, thank you very much. I don't mean to monopolize the questioning here, but
no, it's okay. Great questions. Go ahead.
Just congratulations. I really think that this is an exciting opportunity.
Oh, wow. I really appreciate hearing that. I did hear some criticism from another individual who's I think of as being very knowledgeable, but then I took a little closer look at everything and, well, nah, I don't think so. But any other criticism, like Paul's comment, very, very well said about, well, maybe we need certain levels of things if we are older, but I would look into that and ask why, but it deserves closer evaluation. But anything else, any other criticism?
No, this is sample stuff.
I was just going to say that a company like SomaLogic, the reason why they may not be interested in being a part of what Johnny's doing here is that they're a specialized company. They just want to stay to what they're good at, and that's as far as they go. So they're looking at applying these chips and this ability to get the protein profile of things for various diseases and various things, but they then leave it to other people like Johnny to go further with it and actually apply it to these particular things for therapeutic or preventative or diagnostic or whatever purposes.
Yeah, well, I will say that the several reps I've worked with there have been a couple of changes, but they've been very helpful, supportive, responsive, and they seem, kind of enthusiastic about this concept. And I understand that they're, I think they're good people and that they're scientists, but still they have a business to run. I've let them know that I believe that this will. That I intend to get the word out about, their Soma scan out to our section of the scientific community and let others know about it. So if anyone thinks, thinks they may use this test in their work, let me know and I'll give you an introduction to a couple of people there, and you can carry it from there. So, should we move on to the next topic? I guess so. I had hoped that some of you might know of other companies that I'd mentioned and that are, the true innovators, working on blockbuster therapies. And any thoughts with all that come to mind?
One question is, could this be applied to experiments involving mice, or is you're strictly focusing on humans?
Oh, the protein clock?
Yeah.
Humans only. That's almost my only interest at this point. Mm-hmm. TikTok.
Yes.
So, okay, again, epigenetic reprogramming, gene editing, senolytics, removing pro-aging factors, and mitochondria therapies. Those seems to me like the top ones that we should work on among, dozens of others, but, limited time. We need more time. But, Turn Bio, Life Biosciences, Nugenics, and the others I mentioned, and the scientists. So, okay. I have another thing that I've been working on, age management telehealth physicians. It is actually, it'll be a clinical trial where clients or subjects, patients, whatever you want to call them, will have remote visits with highly informed and trained telehealth, age management physicians via telehealth portal. And it'll be a clinical study, and we're going to collect data. And I created a data management system to facilitate that. I'm going to suggest that I show that to you in another meeting. It's just, we don't want to have too much right now. But so why don't we just move on to an open discussion? What do you want to talk about? You've been very thoughtful to let me hold the floor. There's John's dogs.
Well, I should mute my speaker while he's barking.
No, okay, we like dogs.
So we got phones ringing and dogs barking. Johnny, earlier you asked if I, wanted to ask a question.
Yeah.
So I'm thinking about in the future, maybe not right now because it's just starting out, whether, this could be applied to helping, determine whether somebody is close to the age claimed or not. At the moment, the variability is probably too great to be able to make that assessment. So what in particular, would be the plus or minus? For example, if you had somebody that was 65 years old and you ran these tests, would these tests be able to discern plus or minus 2-3 years how old they were?
Well, I don't think so. And let me show you why. This is something I probably should have covered. So here you see, a roughly linear relationship in this sample file, from the subjects in this sample file that I ran. But so here we see an 18-year-old and a 21-year-old with very low scores. Okay, that's to be expected.
Right.
Okay, here's a 36-year-old who's almost down in that range. And, this person may be practicing a lot of age management and healthy living. So hopefully they will stay biologically younger in later years. And but here we see a 68 and a 71-year-old person down in the lower range, roughly in the range of the 48 to 50 something. So
okay,
I'd suggest they're probably doing something right. Aging interventions, or maybe they just won the gene lottery. But look at this person. Age 44, he's up here in the range of the 70-something. So I've been trying to track this guy down to alert him, hey, get this checked into. But
I bet he's a smoker.
Smoker, probably, couch potato, diabetes, heart, multiple comorbidities in an early age. I don't know, but I would not want to, I'd make some changes if I knew this. So, but this, now that we're talking, this relates to what Paul said about this 36-year-old person who would probably normally be found here. Is he actually less healthy than this person? Is his protein need somehow not being served in whatever it is in his body, whether it's DNA methylation or something going on in cells or something? So that's an interesting question. I hope to find out. I would suggest, well, probably not. He's healthy. I'd suggest probably not. These two guys are pretty darn healthy in their age group, but we would like to find out. That's why taking it to the next level.
I didn't mean to suggest that the person would be less healthy. First of all, that graph is a composite of all their proteins that you've made into some sort of aging clock.
Right.
Okay. I was suggesting that some of their proteins that are in the blood might be higher or lower to affect certain cells, maybe certain tissue types even, that are giving a problem or whatever, or that are aging faster or whatever, you see? So it would go along with John Kramer's idea that we really need in the end the same thing from various tissue types. But of course we can do without that, at least by trying the proteins and see to change some of them and see what happens if we go carefully at it.
Oh yeah, well, yeah, taking it to the next level or maybe a few levels up, yeah, analyzing individual proteins would be a good thing to do. And that reminds me, I've often thought of having this as a personalized method where for an individual person, for their age group or for them individually, analyze which proteins of the most important 9, for example, starting are low in them, and developing a protein cocktail or something that's tailored to their proteome. The ones that are particularly high and potentially resulting in harmful effects, maybe finding a way to trap them and tap that down a little bit. But that's, into the future. Yeah, thanks for clarifying your thoughts on that.
Yeah, I had a comment about proteins. I heard a talk at one of these online conferences recently by a person from the University of Rochester named Vera Gobanova. And she was interested in the question of why it is that, say, dogs live for about 15 years and humans live for maybe 120 if you're lucky, and bowhead whales live for 227 years. And so she did some of her postdocs went to Alaska, where they do hunt bowhead whales, and brought back a whole lot of samples, and she analyzed them. And she found out that there is a particular protein in the bowhead whale blood called cold-induced RNA binding protein, which is CIRBP, that the bowhead whale has expressed about 8 times as much as a human, and it's not expressed at all in mice. And she looked at a bunch of other animals, and there does seem to be a really good correlation between the maximum lifespan and the degree to which this particular protein is expressed. So if you're doing this protein analysis, I think it would be a very good idea to use that one, even if it doesn't correlate all that well with the current age of a person, because it seems to have something to do with the maximum lifespan.
Very cool. Let's dig into that a little bit. And here's the screen. So I'll do a search on
it is CIRBP.
And see if we find it here. Come on, CIRBP. Aha.
There it is.
So I'm wondering, let's see. All right, so there's that. Well, it appears that
looks like they don't measure it, huh? Or else it's not there.
I would have to dig into something. Oh wait, no. Here we are. No, here, better. No, this one. Keep it simple. But Benoit and the team do not use it as part of their calculation, because these are the proteins that they use. So, all right, well, the good news is that we can investigate a little further, and it's already returned as a part of the SOMA scan.
Yeah, I was particularly interested in whether people who belong to these polar bear clubs and go jump in cold water all the time have a longer lifespan than other people, because they would presumably have some of this, more of this protein in their blood.
Uh-oh.
Well, one of the reasons that it may not show up in your 490 plus is that if it is at a certain level, it just stays that level your whole life. And humans may have a lower level than whales or
oh, yeah.
So that could be that it just doesn't vary, you know, in any respect with respect to age.
But the question is, does it vary significantly from one person to another? And does that correlate with their maximum lifespan?
Great question. Great question.
Apparently what it does is it very aggressively repairs double breaks in DNA. And at least some people think that accumulated damage from double breaks in DNA is a determining factor in the maximum lifespan.
That is a good idea there. I just wanted to say, you know, one or two things about the bowhead whale age is I believe they had a situation where they basically took harpoons out of whales that had been harpooned, and then they measured the age of the harpoon. Mm-hmm. The reality is the whales could possibly be significantly older because a lot of the harpooning activity, you know, only started, you know, around the 1790s or so. So we don't know, for example, you know, was the whale 400 years old instead of 220 years old? It could have been much older than the age of when it was attacked, because obviously it must have been pretty big already when somebody tried to kill it.
Right. I see. I was wondering how they determined that, because it doesn't sound like an easy measurement.
One other thing that's interesting with whales is that, you know, obesity for humans and land animals is bad, but obesity for whales is good. And that's partly because the fat is more buoyant than water. So there's actually an advantage to being obese in the water, whereas on land it's a negative.
And it's a good insulator in those cold oceans.
Yes.
I wonder if we should be collecting samples for this SOMA scan from all the super Centenarians that we have the opportunity to going forward. Seems like that data set would be really valuable.
If somebody wants to fund, you know, a super Centenarian study, again, we have no shortage of people that have been willing to volunteer. We actually had somebody volunteer and they died after more than a year because nobody bothered to need them for a study. So I just feel like, you know, it doesn't have to be a huge study. You know, it could be 10, 15, 20, it could be 30 or 40. But, you know, I think that it's really about a matter of funding and vision, because there's number of people reaching 110 is, I would say there's about 1,000 people around the world at minimum. And then you basically about 30% of the data is validatable. So maybe about 300. And then, you know, only 50% are volunteer, but 150, you know, out of 300 could potentially volunteer. I don't think that many would volunteer though, but, you know, to me, I think it would be very easy to get 15.
Does anybody have a library of frozen blood samples from super Centenarians?
Yeah, we actually have some that GRG, you know, we participated in some in the past with the Super Centenarian Research Foundation. That was about 15 years ago. And also Dr. Coles had worked with Stanford, though, more recently. I think it was ended in 2014 when he died of cancer. So I'm not sure exactly what the situation is with that particular situation.
According to SOMA scan, you only need 15 microliters of blood to do their analysis. So maybe one could get some of those samples.
That's a good point, that there's already samples in existence. Go ahead, Johnny.
Oh, no, sorry, I interrupted you, but thanks. I'll suggest, though, with super Centenarians, as you know, if you go to grg.org and look at some of their photographs, they look old. They're phenotypical, phenotypically old. They're often bedridden, quality of life very poor, dementia. So I would suggest that you know, whatever it is about the youthful protein as reflected in, you know, in just by looking at them and their behavior and their mobility has long since gone. And that they have the phenotype and possibly the protein age of a person, you know, that's really pretty old. So I'd suggest that going after their children and asking them for samples, and you know, that would be a better use of limited resources and a better, you know, produce a better measure for our purposes. So Robert and everyone, what do you think of that?
Well, I just want to say firstly is that on the one hand, yes, we do see that many of the children of super centenarians appear to be young for their age and tend to live longer than the average population. However, the limited amount of super centenarian research has already returned significant findings. For example, there was a study of a 115-year-old woman in the Netherlands that found that her blood stem cell lines were nearly exhausted. And that raises the question is, you know, was the maximum lifespan of humans being limited by stem cell exhaustion? Since they only studied one person, that's not a sufficient sample to come to any conclusions. So we're not able to answer that question. Number two, though, they also did an autopsy of her brain, and they found that her brain had no sign of Alzheimer's disease, no sign of amyloidosis. It does seem that amyloidosis and Alzheimer's is a problem. It only affects about half the population maximum and half the population not affected. So I think that, you know, for example, Dr. Coles did his 17 autopsies of super centenarians, and about half of them were dying from amyloid plaques and tangles, buildup, problems that not just in the brain, but also in the heart, the lung, the liver. These are problems that have never been, you know, seriously treated in the past because it was just assumed that these people were too old anyway, why bother? Yet the reality of it is that these problems can already be detected. For example, the NUN study detected these problems already in people in their 20s. So I think we need to have a rethink and stop thinking that, oh, just because somebody's extremely old, that the data can't be useful. This data is already showing that the maximum ages reached are significantly affected by such factors as the genetic protection against amyloid buildup. About half the population has it, half doesn't. And that's just one example.
Yeah. Well, that would be useful. Now, and if anyone steps forward and wants to facilitate that and fund it, Robert is most capable in identifying supercentenarians, navigating, getting appointments and blood draws with them. And he understands the dynamics of the different parties involved, like their physicians and caregivers and others. But, and I would help with that. But, you know, the reason I'm doing this project to take to the next level as a pilot study is for what, in my estimation, is the most important use, which is to compare baseline and follow-up results to measure the effects of the most innovative new aging intervention therapies. And so I would offer it to the researchers and companies working on that. And I just noticed Akshay joined us. Hello. I would certainly want Nugenics and Harold and Akshay to have this protein clock test at low or ideally no cost.
No, thank you very much, Johnny. Yeah, we would be very happy to work with you on that with some of our studies, upcoming studies. You know, protein is actually a very good thing to measure because, you know, we look at epigenetic changes and we look at gene expression changes, but the outcome of all those changes is finally in the proteome, you know. So if we are checking the proteins, I think that's the ultimate. So you are absolutely in the right direction. And hopefully, as the sample sizes increase, the accuracy of your clock will keep increasing. So we would be quite interested.
Well, thank you. I don't know at what point you came in, but I'm going to put a link to a page with a very brief summary.
Johnny, before you go ahead, Dan has had his hand up for a while to ask a question.
Oh, sorry. Thanks. Go ahead. But well, just okay. Just let me finish up. I put a link to the aging metrics, you know, to the protein clock page on the aging metrics website. But there's much more to this. I don't know. What point did you come in, Akshay? What were we?
Yeah, I came in. I came in at 11:30, I think. So I missed that part earlier. But I know that you record these sessions, so I was going to, you know, go and look later.
Right. Well, or you and I, and you know, Harold would be welcome to join. I'd be happy to share what I talked about and more, because I kind of abbreviated it for this group with you sometime. But who had a question? Someone had a hand up for a long time. Go ahead.
Yeah, I did. I hope this question makes sense. Would it be useful to find people who look considerably younger than their biological age and test them? Would that be a useful resource for researchers?
Well, yes, and there are websites now where you take a photo and it analyzes it with AI or something and gives you back, you know, your biological age or your chronological age in my case. It would be useful, but of the thousands of things that are valuable and useful and that we could be spending our time on, we constantly have to prioritize.
Thank you.
Yeah.
Part of the problem there is a lot of people who look younger have been doing special things to their skin, plastic surgery, all sorts of things that make them look younger, but their bodies are maybe falling apart, the rest of them.
Yeah. Well, I'll suggest though something, I mean, if you're really, really interested in that sort of study, that there are other less cutting edge, more immediately available tests that you could do to correlate the two. DNA methylation, certainly, various measures of inflammation, cognitive tests, a functional tests, you know, grip strength, 6-minute walk, 30-second chair stand test, and then various measures of exercise stamina, and there are a few others. So good luck to you if you decide to do that. If you go to the website Aging Intervention and click on Aging Intervention Program, go to Part 4, there are a lot of different tests there that you can consider. Okay. Anybody else? Just jump on in. Oh, Martin's joined us. Hello.
Johnny, I have to sign off. This is Cliff Hagan.
Okay,
Cliff. Thank you again. Great presentation. Without a rock solid metric to measure biological aging, I mean, it's hard to tell whether these interventions do what they claim they're doing or not. And so I really understand why you are laser focused on getting this together. So,
well, just before you go, I'll say, well, thank you very much, you and others, for the encouragement. I'm now even more energized. But I looked at this as a very important measure, but in both the small studies that my foundation does and others I'm involved with, and my own personal age management program, I look at multiple measures of aging. And if most or all shift towards youth after a therapy, well, then I have something, and I keep doing that thing. And I could talk for a while about the different therapies that have been successful and have done that slightly, and others that haven't. But, again, the whole point to taking this this next level pilot study is to really accelerate this with the most innovative therapies that are blockbusters that'll get us a great many more healthy years of life. So thanks, Cliff.
Thank you. Bye-bye now.
Yeah, bye. Well, do we have anything to talk about that would be off the record that I should shut off the recording for? No, I guess not. Well, all right. If no one has anything else and no more questions for anyone, I'll call it a wrap, okay?
Thank you, Johnny.
Yeah, thanks a lot for joining me. And all of us, this is just a wonderful community. Okay, bye-bye.